Good day, students. In today's lecture, we are going to talk about a new topic, that is cell division. So, by now we are familiar with the structure of cell. Now, in today's lecture, we are going to study that how a cell divides or multiplies. Now, question arises, when a cell divides or why a cell divides. As we all know that in a cell, in a particular eukaryotic cell, there is a fixed cytoplasm to nucleus ratio. And when this ratio is disturbed, say for example when the nucleus content is increased, then to maintain the normal cytoplasm to nucleus ratio, the cell need to divide. Now, let us talk about the discovery of cell division. Cell division was first observed by Prevost and Dumas in zygote of a frog, and in case of plants it was firstly observed by Nageli in the plant cell. So, first observed by Prevost and Dumas in 1824 in the frog's zygote. In case of plant cell, as I said you, it was observed firstly by Nageli. And it was in year 1842. Now, before studying the cell division in detail, because there are different types of cell division that we are going to talk about. Before that, let us know about one more term that is called as cell cycle. It is a very important term in the process of cell division. So, first of all, let us know about the cell cycle. As we all know that cycle means some cyclic sequence of events which occur in a predetermined manner. So what is happening in the case of cell cycle? It is the process, or a cell cycle can be defined as the process. So cell cycle is a process in which a series of cyclic changes occur in the cell which leads to its multiplication. So if we write its definition, it is a series of predetermined or programmed cyclic changes by which the cell duplicates its content first and then divides into two or more parts. So, by studying this definition of cell cycle, we must be making two important points. We can make out two important points, that is duplication of the contents of the cell, that is before dividing the cell must duplicate all its content, be it nucleus or the cytoplasmic organelles and cytoplasm. After it duplicates its contents, then it divides into two or more parts. Now, question you might be asking the question that what does it mean that it can divide into two or more parts? As we will be discussing later, that the cell can divide by three ways, that is mitosis, meiosis, and amitosis. So when a cell divides by mitosis process, it divides into two daughter cells. But if it divides by meiosis process, the cell divides into four daughter cells. So ultimately it can be said that after duplicating its content, it can divide into two or more parts. So this point must be clear in your mind now. Now we can represent the cell cycle in a cyclic manner, as the name implies. It is a cyclic sequence of events. Now who discovered cell cycle? The cell cycle was discovered by Palek and Howard. Now let us represent the cell cycle in a diagrammatic manner. It consists of four main phases. If we represent the cell cycle by a simple circle, then we can say half of the circle, that is half part of the circle, is covered by one phase of the cycle, that is known as G1 phase. And on a 24-hour scale, this G1 phase covers half of the 24-hour scale, that is 12 hours. So we can clearly see that the G1 phase is covering half of the half period or half area of the circle, and one fourth phase of the cell cycle is covered by the S phase, and it takes about 6 to 8 hours. Then the next or the third phase is the G2 phase, that covers approximately, or that is completed in approximately. Last phase is the multiplication or division phase, during which the cell divides. And this phase requires relatively least time among the four phases, that is, which means it requires only 1 hour to complete. So we can say this is the shortest phase of the cell cycle. If the question is asked which is the shortest phase of cell cycle, then answer should be the division or the multiplication phase. And which one is the longest phase? Then it should be the G1 phase, because G1 phase requires the maximum time. Now, as you can see in this diagram of cell cycle, we should write it here that it is the cell presentation of a cell cycle. Now, as I have drawn here that there are four phases, G1, S, G2 and division phases. Now, these four phases can be broadly categorized into two parts, that is, the G1, S and G2 phase constitute the long non-dividing phase or interphase of the cell cycle, whereas the division phase is the short phase, and it is also known as short dividing or multiplication phase. So we should write, so the cell cycle is divided into two main phases. The first one is the long non-dividing phase or the interphase. It can be written in short as I discuss. Now this long non-dividing or interphase in turns comprises of the three phases of the cell cycle, that is G1, S and G2 phase. Now, what happens during a non-dividing or interphase? Although the cell is not dividing, but the cell is preparing for division, that is, it is multiplying its content. So, we can say it is a phase of complex changes that occur in a newly formed cell before it divides. So, we should write the characteristic features of the interphase, that is, I said earlier, that this long non-dividing or the interphase occurs in 3 steps. What are these 3 stages? These are G1 phase. G1 stands for the first growth phase. So G1 stands for first growth phase. And during this phase, what are the main changes occur or what are the preparations that cell do before dividing? It is the during the first growth phase, the cell increases in size. For this, there is synthesis of RNA, carbohydrates, lipids, proteins, all the major biomolecules which are present in the cell. They are synthesized and therefore the cell increases in size. Now, the second phase. During the second phase, that is also known as the S phase. S stands for the synthetic or synthesis. As the name implies, during the synthesis phase, the DNA content of the cell doubles. That is, this is the phase of DNA replication. DNA makes up the chromosome of the cell, and chromosomes are present in the nucleus. So we can say DNA is the genetic material and chromosomes contain this genetic material. They are basically made up of DNA plus some proteins. So during the S phase, this genetic material, that is the DNA which makes up the chromosome, it doubles its content by the replication process. So it is the DNA replication phase. So now you have understood the meaning that how the cell prepares during interphase. In the first phase, G1 phase, it has increased in size by synthesizing its lipids, carbohydrate, proteins and all other contents and RNAs. In the S phase, it has doubled its genetic material, that is the DNA content. And now coming to the last phase, that is the third one, that is known as the G2 phase. And G2 stands for the second growth phase. G stands for growth. In some of the books, you will be also coming across the term that is gap phase. G1 and G2 are also known as the gap phases. During this phase, that is G2 phase, the cell finally prepares for the division process, and for this it synthesizes RNA, some proteins that will help in the division process. Besides this, it is the phase during which DNA repair occurs. Although the DNA replication process which occurred in the S phase is a very complex and sophisticated process in which minimum error occurs, but there are some errors which do occur, although they are very less in number, that if there is any error in the DNA replication process, then that error is repaired during the G2 phase. So we can say G2 phase, during G2 phase, proteins are synthesized, RNAs are synthesized which will help in the division process. And also there occurs repair of the DNA which was formed during the S phase. Also, besides this, during the G1 and G2 phase, ATP molecules are also synthesized, because in the cell division process, lot amount of energy is required. So that energy is also prepared during this interphase. Initially, it was believed that interphase is the resting phase because no visible change was could be observed in the cell. But actually, it is the most active phase after the discovery. For the later on discoveries, it was found that interphase is rather most active phase, during which the cell prepares itself for dividing by increasing its content, cytoplasm, nucleus, genetic material, preparing itself for the division by preparing ATP molecule. Now, the second phase of the cell cycle is the short dividing phase. It is also known as the M phase, or the multiplication phase. In short, it is written as M phase. It is the shortest phase. It is the short phase basically because on the time scale of 24 hours, this phase, M phase, requires only 1 hour for completion, whereas the interphase requires approximately 23 hours for completion. And these 23 hours are divided into three different phases. That is, G1 phase requires approximately 12 hours, S phase requires approximately 6 to 8 hours, and G2 phase approximately 3 to 4 hours. Now we'll see that the multiplication phase, during which the cell actually divides into the daughter cells, this phase is also divided into two parts. So firstly we should write that the M phase, during M phase the cell divides. So during interphase the cell prepared for division, and during the M phase the cell actually divided. Now this phase occurs in two steps, or it can be divided into two parts. You can also say as steps. This occur or accomplishes in two steps. The first step is known as karyokinesis. Karyokinesis is also known as nuclear division, and during this phase the nucleus divides. The second part of the dividing phase is the cytokinesis. As we all know that the eukaryotic cell consists of two parts, the cytoplasm and the nucleus. So one part of the cell has divided already, that is by karyokinesis, and during which the nucleus has divided. Now the second part, that is the cytoplasm, which contains the cell organelles, that cytoplasm should also divide into the daughter cells, among the daughter cells. So it is called as cytokinesis. It is also known as cytoplasmic division. So this means division of cytoplasm and the cell organelle. So this is in brief about the cell cycle, which is a programmed, well defined sequence or predetermined sequence of events occurring in a cyclic manner by which the cell divides into two, and before dividing actually the cell prepares itself for the division. Now on this basis, how cell divides, that is on the basis of multiplication phase, the cell division is of three types. These three types of cell division are on the basis of the type of multiplication phase. The interphase is the same among the three types, that is, it comprises of G1s and G2 phase. The first type is mitosis, which forms two daughter cells. The second type is meiosis. This type of division leads to formation of four daughter cells. And the last one is amitosis. It is a relatively simpler method of cell division in comparison to mitosis and meiosis. It also leads to formation of two daughter cells. Besides the difference between the number of daughter cells formed, there are some other differences also, which is between the mitosis, meiosis and amitosis, that we are going to discuss in detail in the later on lectures. That is all for today's lecture. Have a good day.